All right. Looks like we still have people arriving. What we are doing right now is adding another technique to our skill set. This technique is not a huge departure from what we have been doing already. Hopefully, between when you turned in your assignment and today you went and looked at the screencast that I gave you. But just to get us kind of all on the same page, when we looked at the gecko data set, which you turned in on Wednesday, you basically had to combine the population of geckos you were looking at with the season that they were collected in order to make this kind of composite variable that was made up of these two separate things. But in reality, this is one source of variation, and this is a second source of variation. And so it would be nice to be able to analyze those two sources of variation independently of one another, rather than combining them together in this kind of bogus composite variable. And so in order to do that, what we have to do is we have to do a two-way analysis of variance. Now, if you recall, when you first got the gecko data set, there were actually three variables in the data set. There's a third source of variation, which is the type of temperature that was being recorded. And for this particular biologist, knowing whether or not the body temperature differs from the environmental temperature is actually important for the kinds of questions that this person has. So you could just as easily have analyzed the entire data set with a three-way ANOVA. Once you have added a second explanatory variable to your data set, and you know how to do that, you can add as many explanatory variables to the data set as you want. You can have a three-way ANOVA, a four-way ANOVA, a five-way ANOVA, a six-way ANOVA, a 100-way ANOVA if you wanted. Now you would have to have a gigantic data set in order to analyze that, and it would become difficult to explain the results of that sensibly, but it is in theory possible. The math is no different than doing a two-way ANOVA. And so once you expand away from a single factor ANOVA to a two-way ANOVA, it really opens up a lot of possibilities for you. So with that little bit of introduction, do you have any questions from the screencast that I gave you for Wednesday? Did you practice the calculations? Did you go through and do the calculations as you went? Yes.
You just have to do the calculations as like you were doing, right?
Yeah.
Make sure the numbers were the same.
Yep. Yeah. So just check and make sure that my numbers are what your numbers are. If you can calculate it and come up with the same answer that I came up with, we're both doing our math correctly. Yep. Yeah. So once again, let's go back. Well, no, let's not go back there. When we look at this, we have a variety of different kinds of means that we're looking at. We have row means, we have column means, we have the means within each treatment combination, and once again, all of these things are essentially comparing something to a particular type of mean, squaring that difference, and then adding all those up. These are all sums of squares. So in the case of sum squares within, we do sum squares within the same way that we've always done it. We find the mean of male fresh, we find that mean, and the thing that we're comparing that mean to is the individual data points. We do that for male fresh, we do that for male rancid, female fresh, and female rancid, and we add all of that up together, and that's your sum squares within sample. To get total sums of squares, The thing that we're comparing to is the individual data points, and we're comparing those to the grand mean when we're doing sum squares total. So once again, calculate the grand mean, subtract the grand mean from that and square it, subtract the grand mean from that and square it, that from square it, that squared, that squared, that squared, and that gives you the sum squares total. So there's nothing mechanically different about doing a two-way ANOVA than there is a one-way ANOVA. It's just that there are a couple of more means to calculate and sums of squares to calculate because you now have two factors. The one thing is a little different, and I avoided this in the screencast because I thought it would be easier to do it in person, is how you calculate the interaction sums of squares. So sums of squares interaction, which is sums of squares row by C in this thing, the AB. This thing here, what is it basically saying? It's basically saying to calculate this, you calculate the mean of the combination of row and column, subtract from that the mean of the row, subtract from that the mean of the column, and then add to that the grand mean, square that for each of these particular combinations. So, and then what we do is we sum that across all columns, sum that across all rows, and we multiply that whole thing times 3 in this case, which is the sample size within each combination. So what does that mean when we look at this? In the screencast, how did I calculate the interaction term? Yeah.
Wasn't it, you start off with your total sums of squares?
Start off with total sums of squares.
You subtract the sum squares due to the rows, sum squares due to column, and sum squares, which was, that's the interaction sum squares, or the interaction sum squares, that one?
No, this is it. This is. So you take total, subtract the rows, subtract the column, subtract within, and then whatever is left over is the interaction. Yep. But that's not the only way you could do it. That's a relatively simple way of doing it. But another way of doing it is to basically follow the equation here. And so what this equation is basically saying is we have 4 subgroups. We have male fresh, we have male rancid, we have female fresh, and we have female rancid. And for each of these subgroups, we have a subgroup mean. So we have male fresh, male rancid, female fresh, and female rancid. And what this equation is basically saying is you take the subgroup mean, 695.67, you subtract from that the row mean that corresponds to that, which is 615.5, subtract the column mean, which is 669.17, and then you add to that the grand mean, 597.75. You square that whole thing. Then for male rancid, you take 535.33, which is the subgroup mean. Subtract from that 615.5. Subtract from that the column mean, which is 526.33, and add to that 597.75. Square that. For female fresh, it would be 642.67 minus the row mean now changes to 580. The column mean is 669.17, and the grand mean stays the same. Grand mean stays the same. The row mean stays the same. What changes for fresh rancid is the column mean, the actual subgroup mean, which is 517.33, and the column mean, which is 526.33. Square those up, take that thing, and multiply it times 3, and let me know what you get. We're basically calculating this. Yeah.
Like obviously because it's subtraction, like the order matters. So
Yeah, the order for the subtraction, the order doesn't matter, but you're always adding the grand mean.
right.
So this is always the grand mean. This is always the subgroup mean.
So it's subgroup mean minus like the row mean minus the column mean.
Yep. But it could also be column minus row. That what's in the middle order doesn't matter. But you always start with this, subtract row and column, and add grand.
Right.
So an easier way actually to have drawn this up there would have been to put subgroup mean, subgroup mean, subgroup mean, subgroup mean, then the two row means, and then the two column means, and two column means, and then the grand mean. I thought about that about 3/4 of the way through drawing that up on the board. Like, oh, this was easier, a different way. Claire, you're looking confused as if you got a different answer than I got.
Y times 3 is that's the degrees of freedom.
It's the sample size within each of these cells. So remember that you're essentially looking at the subgroup means, and so the thing that you multiply times is almost always going to be what that mean is that you're looking at. Go back to the equation, for example, sums of squares of rows. It's n times q. Q is the number of columns that your row is spanning across. N is the number of observations within each of those things. N times Q in this case is 6, because your row mean in this case is based on 6 values.
Right.
And so you can always think about your weighting on the basis of what is it that you're comparing to the grand mean. This thing has an underlying sample size of 6, so you weight it by that. 918.75. Oh yeah, you sum all that up and multiply it times 3. Yep, yep. And you can multiply each of those times 3 and then sum them up, or you can just sum them all up and multiply times 3. It's the same thing mathematically. Back to your question, when we're doing sum squares of rows, in this row there are 6 observations. Sum squares column, in the column there's 6 observations, so that's what we're weighting by. But for the row by column interaction, the mean that we're comparing is this one, and within this there's 3. Within this there's 3, within this there's 3, within this there's 3.
Right.
So that's where the weighting comes from.
So
Yes, ma'am. Oh, it should be 0.75. Yeah, because these should all be the same. Yeah. Hmm? Oh, was that? Oh, you just copied it down. Yeah, don't ever trust me to write down numbers correctly or write down anything else correctly, actually. This is for the wrong one. So here I did this all in an Excel spreadsheet when I first did this. And what I didn't do was I didn't do the sums of squares of the row by column interaction, but we can do that here pretty quickly. Let me make this big enough that we can actually see it. So what we're basically doing is we're essentially saying, I want the subgroup mean minus the row mean minus the column mean plus the grand mean, and I'm going to square that. Then I want to move to the next one, which is this row mean, this subgroup mean minus this row mean minus this column mean plus, once again, the grand mean, and I'm going to square that. And then I'm going to take this subgroup mean minus that row mean minus that column mean plus the grand mean. And I forgot to put my parentheses, so I'll put my parentheses in there. And then I'm going to square that. And then I'm going to take this subgroup mean, whoop, not all of those, stop it. This subgroup mean minus that row mean minus that column mean plus the grand mean and square it. And then I'm going to add all those up and multiply them times 3. And when I do that, I hope to God that I get 917.75, which I did. Okay, so just like lots of things, there are multiple ways of getting to the correct answer. Whichever way works for you, use that method first. You can implement the formula that is from the table, or you can just add up all of the other sums of squares and then subtract them from the total sums of squares. And it will give you the same answer. In this case, this is what it was for when we have no interaction. This is what it is for when we have an interaction. So I did the same thing in both data sets. All right. The reason that I'm going through this with you is because this is the equations for when we have a two-way ANOVA with replication. We're going to go through, after we finish going through everything with two-way ANOVAs with replication, we're going to go through them without replication. And as it turns out, when there's no replication, this is the formula for the residual term. So we're going to revisit this formula again. I know you're getting a perplexed look on your face, but that's fine. I'll show you why this is the case. The residual term for an ANOVA without replication is not this. It is actually this. And we'll see why that's the case in a few minutes. Well, in more than a few minutes, actually. Any questions about two-way ANOVA with replication? Do you understand how the interaction term works? The first example that we had was a situation where there was no interaction. So this is a situation where the interaction term is really small. And when we run the ANOVA, we find that the interaction between the sex of the rats and the type of fat that they're eating is not significant. When we plot that, we see that fresh fat just leads to more growth in rats. Rancid fat obviously leads to less growth in rats. And the males and the females, they're reacting similarly to the different fat types. So males don't do one thing and females do another thing under a given fat type. They're all doing similar things, but these are doing similarly more growth and these are doing similarly less growth. For the next example, I just flipped them. I flipped fresh and rancid for males. And what we saw is now there's a lot of variation that is accounted for by this interaction. And when we graph that, we actually see that fresh fat affects both males and females, but it affects males and females differently. Fresh fat makes males grow less and females grow more relative to rancid fat, where females grow less and males grow more. This is the nature of interaction. One factor affects the other factor unequally or in different ways. And you will see this when you create interaction charts like this, because the lines will either cross or they will diverge or they will they'll converge. One of these things indicates an interaction. And all it's really saying is that treatment A affects, or treatment level A affects males and females differently than treatment level B affects males and females. Treatment level A affects males and females differently than treatment level B affects males and females. So anytime you have these interaction charts that you make and you see that the lines are not parallel, that's usually some signal that there's an interaction occurring. What's a problem with this data set, just from looking at the plots? Yeah, in this particular formulation, the variances in fresh are much higher than the variances in rancid. We have a sample size of 3. If we did a Levine's test, would we find a difference in those variances? Very possibly not. Does that lack of finding a difference in the variances, is that because the variances are not actually different, or is it due to the fact that something else is going on? Just really too small sample sizes. These variances probably are different, but if we ran a Levine's test on this, we might not find that they are different because we just lack the power to see that. In which case, we shouldn't go blithely on our way. We should stop and examine if there's some way that we can fix this before we actually ran an ANOVA. In this case, if I had a sample size this small, I might not even run the Levine's test to test it. I generally hope that I don't ever have sample sizes this small. I hope I've done my job better than that. Questions about a two-way ANOVA with replication? All right, then let's talk about situations where you don't have replication. So in today's materials on the Moodle site, there is a PowerPoint for two-way ANOVA without replication. Here at the beginning, I'm going to tell you a story, and it illustrates kind of what I'm talking about. Your text, yeah.
Sorry, like this, what you're talking about as far as replication goes?
Uh-huh. Yeah, replication within treatment combination. So here we have, whoa. Here, for each treatment combination we have, male rats being fed fresh fat, we have a replication level of 3.
Right.
Male rancid, replication of 3. Female fresh, replication of 3. Female rancid, replication of 3. There are times when we can't do that, and there are good reasons why we can't do that. And I'm going to describe one real-world example from my own work where we can't do that. So for a number of years, I worked in Puerto Rico, and I worked with a research group that studied the impact of hurricanes on a tropical forest that I used to live in and then went back and worked in. So this is Hurricane George during the first year of my postdoc there, smashing into Puerto Rico. Puerto Rico is just poking out right here. And I worked in a forest on the eastern side of the island. And actually, one of my coworkers, the eye of the hurricane passed right over his house. He was in the hurricane and it was blowing around, and then all of a sudden it got calm, and he and his wife went out and they looked up and it was clear above them and there were clouds all the way around them, and then they could see the other side of the wall coming towards them, and they ran in the house and just managed to close the door as the other side of the eye of the hurricane hit them. Anyway, it smashed into this forest, and generally what happens when a hurricane hits a plot of land is it basically strips all of the branches off of the trees or uproots the trees altogether. This creates canopy openings, opening in the forest. This is a picture of the forest. If you look up at if you just set a camera on the ground, point it up to the canopy with a fisheye lens, this is what the canopy looks like before the hurricane. This is what the canopy looks like after the hurricane. The canopy is largely gone. And so what happens when the canopy opens up is you get all of this material that was up in the trees is now down on the ground. And normally leaves are falling in a forest constantly in a tropical forest. Tropical forests behave differently than our forests do here. In the United States, in temperate regions, you tend to have leaf loss in trees that is seasonal. So fall comes and all the trees lose all their leaves. But in a forest, what happens is trees lose a few leaves each day throughout the entire year. But when a tree loses a leaf, it spends a lot of time before it lets go of that leaf, pulling back all of the nutrients and energy that is in that leaf. So as you see the leaves turning here, what you're seeing is you're seeing those trees pulling back chlorophyll, the photosynthetic pigment, pulling those pigments out of those leaves, storing them in the trunk, and then they will actually reproduce those once new leaves come out. And so the color change that you see is actually a color that was already there. The brown color was already there, but it was masked by the green color of the chlorophyll that was in the leaves. But when you have a hurricane, the trees don't know that a hurricane is coming, and so they can't do that. So all of this leaf litter that falls on the forest floor is full of everything that is normally there, and so they're very nutrient rich. So you might expect that when these leaves get down on the forest floor, there's a lot of energy and nutrients there for organisms to eat. But the other thing that it does is the tree canopy actually kind of forms its own weather. It shades the forest so that it's cooler inside the forest, but it also adds moisture to the air because the trees let loose moisture as they're doing photosynthesis. So the microclimate changes after a hurricane. In the case of the litter deposition, there's more litter and there's more nutritious litter. In Hurricane George, essentially the hurricane dumped an entire year's worth of litter fall onto the forest floor in a 24-hour period. Like, that's how much litter there was. I remember one point after the hurricane, we were having to go back through the forest and remark the trails because you couldn't see any of the trails. And I remember I was scrambling across a bunch of debris over where I knew there was a trail. And when I looked down, I was about 12 feet off the ground because I was just scrambling across broken branches and broken trees. And so there was a trail down there, but you couldn't see it because I was a good ways above where the trail actually was. Microclimate changes result in greater temperature variation and greater moisture variation. It's hotter during the day and cooler at night, and it's generally drier when it's not raining, and it's generally wetter when it is raining. And this is a temperature profile. In tree fall gaps after the hurricane is the red line. You can see at night it's getting cooler. During the day it gets hotter. Night it gets cooler. Day it gets hotter. And then in places where the trees were still intact, you see that it gets less warm during the day. It gets less cold at night. Less warm during the day. Less cold at night, etc. So microclimate changes and total energy changes. This affects thermoregulatory regimes when the climate changes. So I study lizards, and so one of the things I'm interested in is what's the temperature in the forest that these lizards can use to moderate their body temperatures. This is Puerto Rico. I worked in the Luquillo Mountains on the east side of the island here, and our field station was on the northwestern flank of these mountains. Darker green is higher elevation, lighter green is lower elevation. This is where the field station is located. The red represents trails and roads. The blue represents waterways, streams. I study these lizards. These are different kinds of anoles. This is a trunk anole. It hangs out on big trunks down in the understory of the forest. These are twig anoles. They live in the upper parts of the forest, up in the canopy. This is a species that lives in tree fall gaps, lives in sunny spots in what is normally a very shady forest. And this is a grass anole. This is an anole that lives in tree fall gaps after grasses have grown up in those highlight environments. So you can see why these are different species that would respond differently to hurricane disturbance, and one of the things I was looking at was how these guys responded. The problem with all of this is that after a hurricane, all of this stuff happens all at once. The canopy opens, that changes the level of leaf litter on the ground, and it also changes the microclimate simultaneously. And so when we see changes, if we're just following the dynamics of what happens after a hurricane, we can't separate those two effects, a microclimate effect versus kind of this nutritional abundance of the forest effect. And so what we decided to do foolishly was we decided that we were going to make an experiment to try and tease these two things apart. So what we did was we essentially experimentally imposed hurricane disturbance on a forest. So we went out to the forest and we took some plots of the forest and we just kind of left them alone. In other parts of the forest, we basically hired a team of arborists who came in and they cut the canopy out of the forest and dropped it onto the ground just like you would have in a hurricane. And so they used ropes and stuff to get up into the canopy. They sawed the canopy off at a certain diameter of branch and just dropped it down onto the ground. This is similar to what would happen in a hurricane because you have the loss of the canopy at the same time that you have all of this detritus, all of this dead plant material on the ground. that doesn't help us pull these two things apart, these two factors apart. So what we did is we went to another set of plots where we had the arborist cut the canopy out of the forest, but we didn't drop it on the ground. As it came down, we weighed it, and then we pulled it out of that plot. And we carried it to another plot and dropped it on the ground there. And in these plots, we left the canopy intact. So now what we can look at is we can look at the effect of changing the canopy without doing anything to the forest floor, and we can look at changing the forest floor without doing anything to the canopy. So we have two treatments in this case. We have a canopy opening treatment, and we have a detritus addition treatment, and we have experimentally manipulated it such that we have all possible combinations. No trimming, no detritus. Trimming, but no detritus. Detritus, but no trimming. And trimming and detritus. Any ideas on how long it took us to do this and how much money we spent? It took us about 9 months to put it in place, which means it's not exactly like a hurricane, because a hurricane would have done this over the course of a 24-hour period. It took us about 9 months, and it took us about $600,000 to do this. And then we just watched and saw what happened. So it takes a lot of money to do these big kinds of experiments, because we did this on 20 meter by 20 meter plots, and they were surrounded by a 10 meter buffer, 5 meters on each side. And so these are pretty big plots that we're making that all had to be cut. The other thing is we had to find space in a forest that was already being utilized by a bunch of researchers. So what we did was we had 3 different blocks. We had block A, block B, and block C. But within each of these blocks are located on ridges, because ridges are just easier to work on to a certain degree. Also, the vegetation on ridges is generally more stable. But within this block, there's one control, there's one trim plot, there's one detritus addition plot, and there is one trim and detritus plot. In block C, all on one ridge, except that we ran out of room on this ridge, we had to go to the next ridge over. One control, one trim, one detritus, and one combo. Block A, one control, one trim, one detritus, and one combo. There's no replication in any of these blocks. We just didn't have enough money, enough manpower, or enough physical space to put 2 controls, 2 combos, 2 detritus, and 2 trims, or 3, or anything else. These are at different elevations. This is higher elevation, this is higher elevation, this is lower elevation. And so these blocks are unique in some ways, so there's a unique variation that is due to the block, but it's difficult to quantify that variation because we don't have any replication. This is why we have problems sometimes with doing replicated two-way ANOVAs, because it's just physically impossible to do that. And so we need to have a method by which we can analyze these types of data. We monitored these plots for 18 months before we did the treatment, and then for me, I monitored these for 66 months after the treatment, looking at what happened with these animals on these plots. There are other places where we can't do this. If you're a molecular biologist, you oftentimes place your samples in 96 well plates. Well, it might be that this plate is put in an incubator in a different spot than this plate is. As a result, this one has a different thermal environment than this one does. That might affect your results, and it might be nice to know what the effect of that is. In business, you might be looking at 4 different brands of a particular product and how the pricing of those brands varies by supermarket chain. And so here we have 10 different supermarkets. You have the 4 brands, and you have pricing data from these things. Well, obviously, you can only look at brand A in supermarket A once. So there's no opportunity to replicate that. You can only look at brand A in supermarket B once. No opportunity to replicate that. So we see all of these unreplicated designs that are just simply a factor of the fact that we're dealing with things that can't be replicated. We can't replicate William Jewell College. William Jewell College, it is a liberal arts college, but it's different than all the other liberal arts colleges are in some fundamental ways. So even if we were looking at something that varies potentially by college, you could put colleges in groups like undergraduate liberal arts colleges versus regional state schools versus R1 and R2 institutions, but there is a point where something that happens at William Jewell College is going to be different than what happens, for example, at Truman State, which is also a liberal arts college. So we need a way of doing unreplicated two-way designs. So what we have is we have a treatment, and to calculate the sums of squares, we do the treatment much the way we've done the treatment in the past. It's just the treatment mean minus the grand mean squared across all the different treatments. We have a second factor, which in this case, your textbook refers to these as blocks. So this is a table right out of your textbook. The blocks in this case would be the supermarkets. And so once again, what's the average of brand A across all blocks? We would compare those block means with the grand mean and square that. But when you look at the residual value, what you see is that the residual value isn't the same equation as calculating sum of squares residual in the replicated design. This looks more like, well, this is exactly like the interaction term from the two-way replicated design, where you have the individual value of the intersection of block and treatment minus the treatment mean minus the block mean plus the grand mean squared. That's how you calculate the sums of squares residual in this case. What is not in this ANOVA table that was in the replicated design table? Yeah. There's no interaction term. It's impossible to calculate an interaction term because there is no replication. So the interaction term essentially becomes the sums of squares within, and then you have total sums of squares. So how does this all work out? So here are the supermarket data. We have 4 brands of some product across 10 supermarkets. We can get row means, and we can get column means. Column mean would be the average price for product A across all 10 supermarkets, and then the row mean would be the average of this product in supermarket A averaged across all of the brands. And so for each of these things, you could calculate the column means. This is what they are. And you can calculate the row means. Once you have column and row means, you can calculate what the sums of squares are due to brand, and you can calculate what the sums of squares are due to supermarket by simply taking the row mean minus the grand mean squared. Row mean minus grand mean squared. Where's the grand mean? There's the grand mean. So the grand mean here is 2.459. If you take column mean 2.431 minus 2.459 and square that, this is what you'll get. 2.459 minus 2.495, square it, this is what you'll get. So the sums of squares columns is relatively small because each of these is relatively close to the grand mean. But when you look across supermarkets, you see that it appears that some supermarkets have considerably cheaper prices than other supermarkets have. Because there's a lot more variation among supermarkets here, you see that some of these deviations are higher than others. So these are all really, really small. Some of these are bigger. This one is relatively bigger. Plus there's just a lot more of them. Why is that one not any higher than it appears to be? Oh, because that one's close to a whole tenth of a point higher. So many of these are higher than many of these are. As a result, you get a higher sums of squares when you take this mean minus the grand mean squared, this mean minus the grand mean squared, this mean minus the grand mean squared, then sum all of those things up. This is what you get here, and this is what you get here. So we've calculated sums of squares due to row, sum of squares due to column. In this case, sum of squares due to row, 0.1741, it's 451 rather. And sum of squares due to column is 0.05. The degrees of freedom here are, we have 10 supermarkets, so we have 9 degrees of freedom here. We have 4 brands, so there should be 3 degrees of freedom here. We have 40, 1, 2, 3, 4 times 10, we have 40 observations total. The residual sums of squares is the number of rows minus 1 times the number of columns minus 1. So 9 times 3 is what? 27. 9 plus 3 is 12, plus 27 is 39. So our total degrees of freedom should be 39, and 39 is 1 minus the total number of observations that we have. One of the quickest and easiest ways to tell if somebody's doing their ANOVA correctly is by checking their degrees of freedom and see if it is adding up to what it should be. So all we have left to calculate here is we can calculate the sum of squares total, or we can calculate sums of squares within. But sums of squares within, remember, is the sum of the individual values minus the average of the row minus the average of the column plus the grand mean squared. And that's not weighted by anything. Why is it not weighted by anything? There's no weighting in front of this like there is for this and this. For the interaction term, we have weighting here, don't we? What is it weighted by? For the interaction term. It's weighted by n. It's weighted by the sample size within each of those treatment combinations. Why is there no weighting in this formulation? For brand A, in supermarket A, you have a sample size of 1. For brand B, in supermarket A, you have a sample size of 1. There is no sample size to weight by, or there is a sample size to weight by, but that sample size is 1, so there's no point in even putting it into the equation. So how do we do this? Well, for this particular example, we would find the grand mean. So to find the residual term for this example, we would take for the combination of brand and supermarket, brand A and supermarket A, brand B and supermarket A, brand C and supermarket A, and brand D in supermarket A, we basically take the cell mean, I'm going to put this in quotes, minus the row mean, minus the column mean, plus the grand mean, and we take that and we square it. So for this example, this is 2.43 for AA. This is 2.47 for AB. This is 2.47 again and 2.41. This is this first row. So the row means are going to be all the same because they're in the same row, 2.445. The column means are going to be different because we have column A, column B, column C, and column D. So that would be 2.431, 2.495, 2.493, and 2.417. And the grand mean is the same for all of these, 2.459, 2.459, 2.459, 2.459. These are added, subtracted, and subtracted. And you do that for the first row, then you do it for the second row, then you do it for the third row, then you do it for the fourth row, fifth row, etc., etc. That would give you the residual term. This is the sums of squares total. It's just taking each individual value minus the grand mean. So for sum squares total, it's the value minus the grand mean squared summed up across everything. So in this case, it would be 2.43 minus 2.459 squared plus 2.47 minus 2.459 squared plus 2.47 minus 2.459 squared plus 2.41 minus 2.459 squared. That would be it for the first row. The second row would be 2.48 minus 2.459 squared plus 2.52 minus 2.459 squared plus 2.53 minus 2.459 squared plus 2.48 minus 2.459 squared, all the way through the whole data set. When you do that, you would get this big array of numbers. You would add all of those up. So in the end, and this is the calculation for the residual. So you can go through and do the equations yourself and check to make sure that you are doing this. Needless to say, once again, I did all of this in Excel because it just saves some time in terms of making the repetitive calculations. And so what you get at the end of this is you end up with sums of squares due to brand, sums of squares due to supermarket, the residual sums of squares, and the total sums of squares. These should all add up to that. Your degrees of freedom should all add up. There is no interaction term in here anywhere because we don't have any replication to calculate that. So the interaction term essentially becomes the residual term in this case. We calculate mean squared the same way we always have, and then we calculate the F statistic by dividing the brand mean squared by the residual mean squared, brand mean squared by residual mean squared. Both of these things are very large F statistics, and so both of these things are significant. There is variation among brands. Some brands are more expensive than other brands, and there is also variation among supermarkets. So ways in which you could plot this, if you were making a graph, is you can make a graph where you just look at variation among brands. So these are means and 95% confidence intervals. This is the average price of brand A across all supermarkets. This is the average price of brand B across all supermarkets. The average price of brand C across all supermarkets. The average price of brand D across all supermarkets. If you're interested in saving money because gas prices are too high, which ones do you want to buy? You want to buy either A and D. You want to avoid buying B and C because B and C are more expensive. They're not tons more expensive, but they're statistically detectably different in terms of being more expensive. We can also look at supermarkets. This is all four brands averaged for supermarket A. All four brands averaged for supermarket B. All four brands averaged for supermarket C. What supermarkets do you want to avoid if you're trying to save money? ING, certainly you want to avoid ING. They are, once again, statistically more expensive than all of the other supermarkets. You might also want to avoid B, for example. But certainly shop at C or E or J, for example. Although those aren't significantly different from F and H. But there is no interaction. You can't make an interaction plot. That's not to say that there aren't possibilities for interaction, but how do we get a handle on whether or not there are those interactions? Well, one of the things that we can do is we can plot the average value for supermarket and brand. But because there's no replication, there are no averages, which is why there are no confidence intervals, because you need a sample size in order to calculate a confidence interval. So this basically is just showing how brands A, B, C, and D vary across the supermarkets. What do you notice about this figure? Somebody other than Karis. He's done all the talking today. Cole, what do you notice?
Well, I mean, it looks like they follow the same trends, just at different prices.
Yeah, so we said that B and C tended to be higher than A and D, and B and C is higher than A and D everywhere, right? These prices are changing from supermarket to supermarket, but they're generally changing all in the same way, aren't they? Very little crossing of the lines, and when the lines do cross, the crossing is relatively minor. Basically, when a supermarket is more expensive, that supermarket is more expensive for all brands, and the order of those brands doesn't generally flip around very much. Prices are low, they're similar to when prices are high in terms of the ordering of things. Prices low, prices higher, prices lower, prices higher. Sometimes the magnitude of the difference between the high and low prices is this here and is slightly higher here. But generally speaking, these brands are moving in parallel with one another. So even though you can't statistically test whether or not there is an interaction, you can produce a graphic that will give you some hints as to whether there is an interaction. In this case, there really isn't an interaction. What happens to one brand happens to all the other brands as they vary across the supermarkets. So you see these things all moving in parallel. If there was a lot of crossing of these lines such that things weren't moving in parallel, you would probably conclude that there was an interaction, even though you can't statistically test that interaction. So there are ways of using visualizations to tell you more information about your data than a statistical analysis will reveal. It's just that this is something you can't statistically test. We're not done with two-way analysis of variance yet. There are some other things that we need to talk about back with the two-way ANOVA with replication, but we will cover that on Monday. Questions? All right, then I will see you on Monday. Yes, ma'am.
Find out what the skill was that that little blue team.
I did. Yeah, it was a raccoon. Oh my gosh, he was a handful. Yeah.